So thanks everyone. Just remember now to mute yourselves and put your cameras off. I'm just gonna unpin Savarna. And yeah, so it gives me great pleasure to introduce our first speaker, who's definitely been a massive stalwart in the botanical circles, Dr. John Manning, who works at the Compton Herbarium. And I went out and got myself something. So Dr. Manning has written many books and publications on the Cape and South African and Southern African flora. And we're so thrilled that he's made the time to come and talk to us today about these latest publications. So John, if you could share your screen on that side, and we look forward to hearing from you. And thank you for your generosity.
Hi, Rupert. Can you hear me?
Yeah.
Hello, everybody.
And PowerPoint is showing fine.
Great. You know how I hate public speaking, so this is a miracle that you and Ishmael have managed to get this. It's wonderful to be here with everybody, and you waving that huge book around. I've got to tell you that literally 10 minutes ago, the Saunders Gladiolus book was dropped. My copy was dropped off here at the herbarium, and it's very, very beautiful, people. So save your money for Christmas presents. I know Botsock is hosting the launch in 2 weeks' time. Is that right, Rupert? The 26th?
Yeah. So we'll all be involved. So everyone must keep an eye on the social media.
Wonderful. Well, it's really beautiful. Great. Tonight's talk is, Ishmael asked me to share with you some of the findings, general findings and news about this Iridaceae monograph. So I wanted to run through with it first. What actually is it? So it's essentially the first complete account of Iridaceae of Southern Africa since 1896, when Baker did the treatment for Floricapensis. The species circumscriptions in the book are informed by extensive field and laboratory study over combined 60 years. That's not 60 years of my life, that's 30 years of mine and 30 years of Peter's, basically. Another significant part of it, which dawned on me, is that the taxonomy is based on a consistent philosophy throughout the whole book. So throughout the whole family now, we have a consistent philosophy. That's Peter and mine's. That's not saying it's the right one or the only one, but at least it is consistent across the whole book. It also includes new classification based on current latest molecular phylogenetic data. And then obviously the other normal stuff in a taxonomic account, identification keys, descriptions of all the taxa, maps, and a complete synonymy, which has got thousands and thousands of names in there. And the numbers, it's A4 format. It's huge. As you saw, Rupert was holding this thing up. It's big enough to drink your coffee off. It's 1,159 pages. It covers 1,210 species. We have a drawing for each genus and then color photographs for almost half of the species. So what does it look like? It's that huge thing there on the middle of the screen, placed on some of the other books we've done to give you an idea of the size of it. And then if we open the first account, you'll see there it's got very nice layout that was done by Pretoria. Family account, keys to the subfamilies, descriptions of the subfamilies, genera, little story about them. And then there you'll see the drawing for Aristea, showing characteristics of Aristea, these keys to the subgenera if they are, or sections if they are, keys to the species. And then if we look at one of the accounts, there's a map for every species with little dots and the normal stuff, description, synonymy, and some little ecological information about it. And then at the end of each species, each genus section is these combined photographs, which, as I say, in total illustrate about half the species. Now, general discussions, well, facts that emerged from doing such a monograph of a megadiverse family Just to put it in context, the family includes an estimated 70 genera and something over 2,160 species worldwide. And slightly more than half the family, that's 36 genera and 1,210 species, occur in Southern Africa, which makes the subcontinent a major center of diversity for Iridaceae. And then on top of that, and as you all know, being Kru people, most of the Southern African species are endemic here, especially in the winter rainfall belt in the southwest. Although the Drakensberg escarpment of KZN and Lesotho is a secondary center of diversity, and I'm sure we'll hear more about that from Ralph. Now, in general, the development of systematic study of Southern African irids, the family has benefited from extended taxonomic work, and that initially was based in Europe, but then later in South Africa. And I must highlight the pioneering work by Joyce Lewis, who revised Babiana, Gladiolus, and Therianthus in the 1940s through to the 1960s. And then Miriam de Vos, who was based at Stellenbosch, and she did Romulia, Syringodia, Tritonia, and Tritoniopsis in the 70s and 1980s. And then the father of Southern African Iridaceae is, of course, Peter Goldblatt, who started working on the family in the 1980s. So you'll see there's been a succession of people who've been dealing with the taxonomy of the family. So as a result of these, essentially these three authors, 437 species were recognized in the century following Baker's treatment in 1896, which essentially doubled the number of species in the family. And then after I started working with Peter in about 1990, we increased that number by another almost 300 species in those third of a century. So there's the summary stats. Baker 1896 recognized 437 species. By the time Peter Goldblatt's summary was published in 1991, it was up to 917. And in 2020, it was up to 1,210 species. Now, this looks a bit daunting, but this is, I find this very interesting. We can break down the study of Iridaceae in Southern Africa into three main phases. And the first phase, which culminated in Baker's work, relied solely on the morphological examination of herbarium specimens. Obviously, the scientists were based in Europe, and that's all they had recourse to, apart from species that were in cultivation, of which there were quite a few. But basically, they were relying on herbarium material. And on top of that, they placed special emphasis on the structure of the flowers, particularly the symmetry and the fusion of various floral parts. And this is a direct result from the central role that Linnaeus allocated to flowers in his classifications. And following the taxonomic principles that were in operation at that time, Baker, as I say, recognized 437 species of Iridaceae from Southern Africa, which he distributed among three, what he called suborders, which we now call subfamilies, which he defined by the structure of the inflorescence. And the largest of these, which was the suborder Iridaceae, which is now subfamily Crocoides, he defined the genera primarily by the division of the style branches and the symmetry of the flowers and the length and the shape of the floral tube. Now, I highlight that because recent phylogenetic studies, that's evolutionary studies with DNA, have demonstrated that the inflorescence type and the style branch division are indeed very informative about evolution in the family. And we still use them today to diagnose the higher level taxa, that's like subfamilies and genera. But in sharp contrast, the other set of characters that Baker used, which was the length and the shape of the floral tube and the symmetry of the flowers, has turned out to be almost useless. Those characters are, we now understand, are very susceptible to evolutionary change through pollinator-driven influences. And we only realized that when Peter and I started working on the plants extensively in the field, and then the molecular data could allow us to interpret that. So you can see how the source of the data that one has will influence one's understanding of these things. So after Baker's work, the second, what I call the phase 2, which is the morphological cytological phase, which took up that century after his work, it stemmed from the essentially the shift of the study of the family here to Cape Town with the development of departments of science at the University College of Cape Town, is now UCT. And coupled with that was a huge rise in local interest in the flora and the development of networks of contacts among surrounding countryside, farmers and landowners. And that was through the work of Louisa Bolus, who sent her students and her colleagues out and did a lot of outreach, what we call now outreach. She wrote all sorts of popular books on South African plants, and she really encouraged an interest in them among landowners, which was a first at the time. And in fact, any of you who've worked on systematics in this country will know that many of her new species were published in what is essentially the equivalent of house and garden type magazines. They were not in scientific journals. So when you were reading about chrysanthemums or anthuriums for your garden, you would also encounter all these new species. So on top of that, the emerging influence of evolutionary thinking can be seen in these revisions over this period by the way that the authors have arranged the species from the first ones to the last ones in any of the genera. And so generally they had short tube symmetrical flowers in the beginning, and then going on to species with longer tubes or zygomorphic flowers. So you can see the same kind of thinking that Baker was using. And also at this time, there was a huge interest in chromosome numbers. And that was because at the time, and particularly in America, botanists were starting to use this notion of reproductive isolation as a species character, meaning species can't interbreed. That's essentially the kind of thinking that they were following. And so there was a lot of interest in chromosomes as chromosome number and chromosome behavior, and in hybridizing and things like that. So that period from the 1940s through to the 80s, it saw the publication of many of the groundbreaking generic revisions in Iridaceae, to the extent that by the time Peter summarized the knowledge in 1991, he felt confident enough to state that, or write, the basic taxonomy for the family is comparatively well known, although new species continue to be discovered and some changes in generic circumscriptions are likely as our perception of phylogeny and relationships change. So what that really meant was that there was some form of taxonomic revision published for all or most of the genera, and that in consequence, all or most of the specimens that were coming in could be accommodated in the known species, sort of more or less satisfactorily. Well, we only know now how wrong that assessment of the situation was. And at this stage, Peter also was a pioneer with this cladistic morphological study. And so he applied that and he got a new classification. And as I say, the number of species had doubled since in the last 100 years. So now we come to the final phase, this ecological molecular phase, which is the last third of a century. And that really owes its existence to the two new sources of data. This was our intensive field study of pollination biology, and that really was a fluke, really, because I studied entomology in my undergraduate years. So I was very interested in insects and applied that to now the Iridaceae, and it was the first in many ways. And so we did a lot of pollination studies. We got to know the species really intimately in the field, and we really understood for the first time how important these different pollinators were in driving the flower form in all these genera. And then on top of that was the emergence of the DNA molecular techniques that provided an independent data set. Now, what I mean by that is that it's a data set that's not dependent on the morphology or the pollinators. It's driven by different sets of influences. So it's independent from the morphology to many extents. So it's a really nice way of testing the morphological stuff. And so we had that new set of data. And so to summarize then, we've used the molecular sequence data to investigate the relationships among the genera, but we haven't yet used it to test whether the genera are natural as we are currently understand them. So one of the findings was that the DNA-based classification is only partially congruent. It only partly matches the morphological one. I'll show you a table in a moment. While both of them retrieved the subfamilies Crocoideae and Iridaceae, the morphological classification failed to recognize that what was then the Nivinoideae was actually two different subfamilies, okay? And the morphological classification was also unsuccessful in resolving the relationships among some of the genera of the Iridaceae. So what we could do then was we removed Iridaceae from the Nivinoideae to make a subfamily by itself. We moved Palansia into Watsoniae tribe because it had previously been put in a tribe by itself. And we also took a Tritoniopsis out of one of the tribes as a separate tribe. So we could tweak some of these relationships, but by and large, they matched quite a lot. And then finally, Bobatia was kind of thought to belong to South American genera of Iridaceae because of its morphology, but actually the molecular stuff confirms it's wrong. That was wrong, and in fact, it is a Southern African. It's a Southern African thing. So this is just summarized for you guys to have a look at. On the left is the morphological classification. Far left column has got the 3 subfamilies. And then the second left column, you'll see the Ixioids. There were 3 tribes. And going down to the bottom on the second left column, the Iridaceae had 2 tribes. So that was the morphological classification that we had in 1991. Now, if you go into the right-hand pair of columns, you'll see on the far right, Nivinoideae has been now split into 2 subfamilies. Iridaceae is a subfamily by itself. Otherwise, the other 2 match exactly. The names are different, but that doesn't matter. And then if you go into the tribal level, on the right-hand side, you'll see Tritoniopsis has been pulled out as a tribe by itself, and Palansia has been included with Watsonia. And then if you go at the bottom, as I said to you, Bobatia, which was in the Scissorinki, has now been moved in with the other Southern African genera. So there's been a bit of refining done there. And to summarize this now, the increased field study, and I can't stress enough how important the field study was in enabling us to understand these things, combined with the recognition of the importance of ecological characteristics like soil type, modes of vegetative reproduction, that's whether they have stolons or cormlets or whether they don't, and the pollination biology, how important those factors were in driving in speciation. We could refine our understanding of what the species are and recognize those 293 additional species. So in summary, then, the species, we still define them by morphological features. We just don't have other data for the bulk of them. So we still define them by what they look at. But because we know the variation pattern so well in the field, because of the amount of field work we've done, and because of the amount of field work that you people have done by collecting specimens from areas we've never been to and will never get to, and sending photographs, because of all of your work, we really understand now what these things look like and how they vary in the field. And you'll know that Ishmael, after this book came out, the year after this book, in fact, the year it came out, I think, Ishmael found another new Morea. it's one of 3 new species that have been discovered since the book comes out, but all of them are really, really localized endemics. Okay, so I don't anticipate we're going to find very many more, and if there are, they're going to be very local endemics. So finally, Peter's observation from 1991, we can repeat it exactly now 30 years later. The basic taxonomy for the family is well known, although new species continue to be discovered, and some changes in generic descriptions are possible as our understanding of relationships improves. So we say the same thing, although we now know that 30 years down the line, Peter was a bit faute of devoir to say it then, because it was shown not to be true. And what we do understand now, however, is that that kind of sentence or claim really depends on the level of understanding that exists at the time. So while he could say it at the time, he was unaware that a lot of understanding was inadequate. And so therefore, we can more accurately say, and this is going to sound very fancy here, more accurately, we can only say that our knowledge of organismal variation, that's the variation in the family, it's increased to the level at which we now have a workable morphological description of the variation, which will accommodate the great majority of specimens. In other words, we can put a name on most of the specimens, nearly all of them, in fact. And we can do that without disrupting the species as we understand them at the moment. So what this suggests to me is that major changes in the taxonomy are going to require a whole lot of new sets of data and appreciation of what the species are, because I think we've gone as far as we can with the current way of defining species. And so any big changes are going to require a new way of understanding species, and that's going to be DNA-based studies, if they're ever done at all. And that's necessary before I think we overturn anything. So we'll see in 30 years' time if someone says that was true or not true. Summary statistics, just briefly, to just show you that this book didn't appear out of nothing. In the last 30 years, we published 20 popular articles, 28 books or chapters, and 133 scientific papers that all led up into this book. So these things require a huge amount of preparatory work. And then this is just to show you quickly what our phylogeny or our phylogenetic evolutionary tree is for the family. And that allows us to make some claims that really significant innovations in the family were the evolution of the corm, which actually evolved twice, both in Southern Africa, and both times at about, we estimate, about 30 million years ago. So for the first time, we can propose a date for this and actually identify that it happened twice. And then the second innovation is the fact that the inflorescence changed from a cyme, which I'll tell you about now, to a spike, which we speculate allowed the flowers to be presented horizontally rather than vertically. And it allowed the evolution of zygomorphic flowers, that's irregular flowers. And the result of this was that the pollen could be placed for the first time precisely on a particular part of the pollinator rather than just everywhere of the pollinator. And this spike evolved only once in Southern Africa, a little bit later than the corm. So there was a lot happening in that period, 30 to 40 million years ago, we think. Okay, don't spend a lot of time on this. What I'm just wanted to put this up for you guys to see, ladies to see. The genera marked in red, that's Iridaceae, Tritoniopsis, Tritonia, Renown, and Barbatia. We reanalyzed those genera for this book. So we revisited the taxonomy of those genera because they were genera we haven't done. And so the book includes new taxonomic understandings of those genera. All of the other genera we had looked at ourselves. So those were the ones that we hadn't looked at And so we could reinterpret them to fit in with our understanding of taxonomy so that we got this consistent taxonomic understanding. And then, as I promised you, just to alert you, the two main inflorescence types in the Iridaceae. On the left-hand side is what is this thing called a cyme, and that's in Morea and Iridaceae. You get all those flowers pushed into those little sheathing bracts. They all come out like in a clump at the top. You'll see the flowers have to face upwards because they squeeze up like out of those little sheaths like toothpaste out of a tube. And then in contrast, we have on the right-hand side a hesperantha with a spike, and there you can see this, the individual flowers, solitary flowers, are arranged up the stem. One flower in each of those little bract clusters, whereas on the left there's several flowers. You can see their old fruits there. So they're quite different inflorescence types. And that's what we mean when we say a cyme and a spike. And this is just to indicate, don't get caught up in the details. On the left are the main characters that are used for the subfamilies, and across the top are the subfamilies, and you'll see each subfamily is defined by a combination of inflorescence type, whether the flowers have little pedicels or stalks or not, the symmetry of the flowers, whether they have a tube or not, whether there's a corm or not, the length of the style branches, and then whether the leaves have a midrib or not. So those kind of basic 1, 2, 3, 4, 5, 7 characters in different combinations are enough to recognize the 4 subfamilies. And then Ishma asked me just to run through the main groupings with you. So we're going to really just be doing subfamilies here and tribes. So Iridaceae, they're evergreen. The rootstock is a rhizome, no corm. The inflorescence is this cyme, in other words, lots of flowers or more than one flower from one little cluster. The flowers are short-lived. They last less than a day in nearly all the species. And they are symmetrical, and the tepals are free, and the little stigmatic branches are very short. So those are the characteristics for Iridaceae, and they include just this one genus, Iridaceae. You can see there at the bottom right, the little rootstock is a rhizome. There's no corm there. Flat leaves without a midrib. The little cymes with several flowers in them, very short style branches, and the symmetrical flowers that last only a few hours of each day. Then the second, so that's a very small subfamily, one genus. Then we have Nivenoids, and this is essentially only for people down here on the fynbos. They're shrubby. They really are shrubby. I think, Rupert, didn't your daughter climb into one once? I think they're big enough to have children climb into them.
Not yet.
Not yet. Inflorescence is again the cyme, but the flowers are sessile. In other words, they don't have a stalk. And again, the corolla is regular or actinomorphic, but in this case, we actually have a tube. And so here in this Nivenia, you can see a very nice floral tube. And there are 3 genera, and we won't go into any more details about them. They're all restricted to the mountains here in the southwestern Cape. Then the Iridoideae, which is a very big subfamily. They're evergreen or deciduous herbs. They have a rhizome or a corm. But the corm is a very particular kind of corm. Okay, it's only 1 internode thick, long, and it's axillary. Now, what that means, and just bear with me now, if you think of a garden gladiolus, I think all of you have planted a garden gladiolus or a garden sparaxis or an ixia, one of those things. The corm, the swollen base of the stem, is developed literally at the base of the leafing stem that you see emerging above the ground. The base, the basal internodes swell up. And so that corm is formed from the base of the actual leafing stem. In this lot, in these moreas, the new corm is formed from a little bud at the side of that stem. It doesn't look like that in the adult plant because it's all covered with that fibrous tunic. So it looks like the flower stem is coming out of the top of a corm, but it's not. If you open those tunics, the new corm is sitting at the side of the stem. So it's a completely different formation, all right? So these have these, what we call auxiliary corms. Then, for instance, is a cyme, as I showed you in the earlier picture, the yellow morea. Flowers are pedicellate. They're also often short-lived, and they're always symmetrical. The tepals are usually free, and the style is deeply divided in most species with those nicely wide, flat, petal-like styled branches. Okay. And these are the genera in that subfamily. With Bobatia, Dietes, those are the two genera with rhizomes. They don't have a corm. And then we have Ferraria and Morea that do have corms. And then the last subfamily, which is the very biggest here in Southern Africa, is the crocoids. They have a corm, but the corm is several internodes long, and it's formed from the base of the leafing stem, unlike the Morea type corm. And the inflorescence is a spike. You can see that nicely on that hesperanthe on the left there, although sometimes those spikes are reduced to just one flower, like in Rhombulia. Flowers are sessile, and here the corolla can be regular or irregular. The tepals are always joined in a tube, although it can be very short. And the style is deeply branched, but the branches are very slender. And so in that big subfamily, we have 3 tribes. So Tritoniopsis, which again, sorry, people from upcountry, it's entirely a greater capitalistic region genus. Very interesting genus. They have these odd corms, multiple main veins on the leaves, details of the bracts that you can see there, and other little odd details of them. The stamens, they often fold back. So there is Tritoniopsis. Lots of veins on the leaves, and the corm is that strange type, although, as I say, you can't see it because it's covered with these tunics. So that's a very interesting genus by itself, in a tribe by itself. Then we have tribe Watsoniae, which has got quite a lot of little genera in it, and usually it has deeply divided style branches. That's an easy character for it. And those are the genera there. If you have a look at the top left-hand corner there, drawing of Watsonia, that's the little style, tip of the style. You'll see it's divided into 3 branches, and each branch is then deeply forked. So they look like 6 little style branches there. So that's a character across this whole tribe. And it includes the genus Watsonia, which, of course, there are lots of those upcountry. Palansia, it's only here in the Hottentots, Holland. Therianthus is only down here. Micranthus is only down here. And then we have Laperosia group, mostly down here, but also quite a lot up in Namibia and going into tropical Africa. And then the final tribe is this Ixie, and this has this axial corm development that I was telling you about, like in Gladiolus, with the base of the stem swells up. And we have lots of sub-tribes in there. Now, initially in Peter's classification, Rupert, how much, what's my timing here?
You've got at least another 15, John.
Okay, fine.
Yeah.
In Peter's original classification in 2010, I think, he divided this the Ixie, this tribe, into the crocoids into, I think, about 7 tribes. But the problem was that was based on the DNA phylogeny. But the problem was that you couldn't recognize them. There were no characters that define, morphological characters that defined each of the tribes. So although they represented real clusters of related genera, we couldn't recognize them. You just had to go on the fact that they were related. So we then decided rather that in this new IRID book, we'd put all of those into that tribe Ixie, and we'd have lots of sub-tribes that you could at least recognize. So you'll have to bear with me now. We're going to go through quite a few of these, but at least it'll give you an understanding of what the genera are and how they're related to one another. So Gladiolus subtribe, it only includes Gladiolus. It's quite distinct from all the other genera in that group. And it's difficult to define some of these genera, I must say. So Gladiolus, fine, they're unique in having those disc-like seeds. They're flat, papery seeds with a wing. You can see it's in the middle top line there, that funny little flat thing. It's like a discus. So the seed body is that round thing in the middle, like a little pea, and then there's this papery wing around it. But if it doesn't have seeds, it's not really very useful. So combined with that, the style branches are widened, which again is a little bit iffy. And then significantly, that on the top line then, the red, X equals 15. X is the abbreviation or the code for the basic chromosome number. So in Gladiolus, the basic chromosome number is 15. In other words, you'll get multiples of 15 in the species, and that is very unusual. So all the Gladiolus have the same chromosome number, which is why they hybridize very easily. Okay. But X, the base number is 15. You'll see when we go down to the next subtribes, they differ from that. So there's Gladiolus all by itself, quite a distinctive thing. Then we have little Melis ferula, and again, that is a winter rainfall thing. It's also very distinctive. It's all in a little subtribe by itself, a very strange looking thing with its highly branched inflorescence, these strange, unique bell-shaped corms that split in a particular way, and these strange little three-lobed fruits. You cannot mistake it for anything else. And you'll see there then on the right-hand side on the third red line, the basic chromosome is 10, which is much more common. So 15 for Gladiolus was unusual. So this is Melis ferula. Again, it doesn't have any close relatives. Then we have a group of 4 genera that are very closely allied, and they include Frisia, Crocosmia, which is essentially upcountry, Devia, which is only from the Roggeveld escarpment, that's near Sutherland, and Xenoscapa, that's again a winter rainfall genus that extends into southern Namibia. And they again have notched or divided style branches like we saw in Watsonia. So you see again, these characters can reoccur independently of one another, which is what led to a lot of confusion among early botanists who didn't have any evidence for this, so they assumed that it was shared character. And then you'll see the top left column, left point there in the red. They have a particular anatomy of their leaf margin. So I'm highlighting these characters just so you can see the kinds of characters that one uses, and you can't tell up front what the characters are that are going to be useful. You have to just study all of them, and then some will emerge as being useful or not. And so we have Subtribe Tritoniinae, which has these 4 genera in it. And they have nice glossy seeds, which is another good character for them. Then we have Subtribe Hesperanthinae, which includes just these 2 genera, Geiseriza and Hesperantha. Again, you'll see the base number is 10. And they have always actinomorphic flowers. They have that strange leaf anatomy, and they have asymmetrical corms, either asymmetrical or bell-shaped corms. So then they're not round globose corms like Gladiolus, for instance. Now, interestingly, in these 2 genera, we don't have a proper understanding of the relationships among the species. So I think it's very likely that Hesperantha is derived from Geiseriza, and so Hesperantha, the genera those 2 genera may not be natural genera, what we call natural genera, in other words, real genera. The Hesperanthas may be specialized Geiserizas, and I think Evan Eifler was looking into that, but we don't have any answers from him yet. So that is likely, I think, going to be a source of problems with the names, because it may turn out that those 2 genera have to be combined. Then we have Subtribe Tritoniinae, which again, they have those nice smooth glossy seeds. And a very strange feature with them is that the little vein in the seeds is often floats free from the seed body. So again, that was a character that we discovered, but we had no idea what it really signified or why that even exists like that. But it does, and it seems to indicate relationships among these genera. And there's 4 plus genera. There's Babiana, which is a big genus, 93 species. Casmanthe, 3. Sparaxis, we all know, there's 16 of them. Dathiastrum is the upcountry genus from the center of the country, Kimberley, the basin of the Vaal and the Orange River, really. Very strange thing. So those 4 genera, plus Diorama, which upcountry, Tritonia, which is countrywide, and then Hexia, which is basically here in the Cape. So all those 7 genera are in that subtribe. Then we get another weird little genus by itself, Renown. It's only known from the Eastern Escarpment in Mpumalanga and in Limpopo, and then it extends into tropical Africa. They look like little gladioluses, and initially we assumed that they were closely related to gladiolus. Particularly, you'll see they have X equals 15. So they have the same chromosome number as gladiolus, but they're not related to gladiolus. They have normal seeds, but other than that, they're kind of difficult to define. So there again is another example of how these characters that you think are indicate relationship, like in this case, basic chromosome number, turns out probably doesn't. And then we get, I think this is the last subtribe. This is the kind of stemless ones. They all cluster together. You know these genera, Romelia, Afrocrocus, and Syringodia. They all tend to be stemless. They have these woody corms, often asymmetrical, and these symmetrical flowers, which may be the result of the fact that the inflorescences are reduced to single flowered little branches and the flowers stick straight up, so they can't really be anything other than symmetrical. And then they have this rather strange chromosome number. We think it might be 14. And I think that's it. Yes, that's the end. Thank you very much. Does anyone have any questions after all that barrage of information?